You will hear a two-part lecture about CRISPR, a revolutionary gene editing technology. You will hear the recording once only. Before you listen, read the questions carefully. Now, you will hear the first part of the lecture, which focuses on the discovery and technical aspects of CRISPR. Complete the sentences below.
Good afternoon, everyone. Today we'll explore CRISPR, a groundbreaking tool in modern genetics. CRISPR, short for clustered regularly interspaced short palindromic repeats, was first identified in bacteria in the late 1980s. At that time, scientists didn't realize its full potential. They thought these repeating sequences were simply genetic leftovers. It wasn't until 2012 that researchers Jennifer Doudna and Emmanuel Charpentier discovered CRISPR could be paired with an enzyme called Cas9 to cut DNA at precise locations. So, how does it work? Imagine the DNA as a massive library of instructions. CRISPR acts like a GPS system, guiding Cas9 to the exact page in the genetic book. Cas9 then makes a cut, and scientists can either delete unwanted sections or insert new ones. The precision of this process is what makes CRISPR revolutionary. It's faster, cheaper, and more accurate than previous gene editing techniques like zinc finger nucleases. Applications are vast, from correcting genetic mutations responsible for inherited diseases to creating crops that resist pests and extreme weather. However, remember, accuracy is crucial. Even a small off-target cut can have unintended consequences, which is why research is still ongoing to make the technology safer.
You now have 1 minute to check your answers. You will now hear the second part of the lecture, which discusses ethical and future implications of CRISPR. For each question, choose the correct letter A, B, C, or D.
Now, let's turn to the ethical dimensions. While CRISPR could potentially eliminate diseases like sickle cell anemia, it also raises serious concerns. One of the most debated issues is germline editing, modifying DNA in embryos so that changes are passed on to future generations. Some argue this could prevent devastating illnesses. Others fear it could open the door to so-called designer babies, where parents select traits like height, intelligence, or even eye color. Another challenge is accessibility. Will CRISPR treatments be available only to the wealthy, increasing the genetic divide, or can they be made affordable worldwide? Then there's the question of regulation. At present, different countries have vastly different laws. Some permit research on embryos up to a certain stage, while others ban it entirely. Looking forward, many scientists believe CRISPR will become a standard medical tool within the next 2 decades, provided we develop robust safety protocols and international guidelines. Whether this future is utopian or dystopian may depend less on the technology itself and more on how responsibly we choose to use it.
You now have 1 minute to check your answers.